we are discussing about the Gullian Barre syndrome pathogenesis. What is the pathogenesis? What pathogen cause the Gullian Barre syndrome, polyradiculopathy? 70% of cases of GBS occur 1 to 3 weeks after the respiratory or GI tract infection. 70% of GBS cases occur after the respiratory tract infection, after the respiratory tract infection, or after the GI tract infection, gastrointestinal tract infection. 70% of the cases of the GBS occur after the respiratory tract infection or GI tract infection. This GBS occur after this infection 1 to 3 weeks after. 1 to 3 weeks after this infection, the GBS symptoms in GBS can occur. 20 to 30% of all cases in the North America of GBS reinfection with Campylobacter jejuni occur. And 20 to 30% of cases of the GBS, 20 to 30% cases of the GBS in North America, the reinfection with Campylobacter jejuni occur. Reinfection with Campylobacter jejuni. After the infection of the Campylobacter jejuni, there is reinfection of the Campylobacter jejuni occur in 20 to 30% of the cases, and these cause the GBS in the North America. Reinfection due to Campylobacter jejuni can cause the Gullian Barre syndrome. 70% of GBS occur due to after 1 to 3 weeks after the respiratory tract infections and GI tract infections. Other infection can also cause the GBS. Example of other infection is cytomegalovirus, Epstein-Barre virus anomaly, Epstein-Barre virus, cytomegalovirus, HIV virus, hepatitis E virus, hepatitis E virus, and Zika virus, hepatitis E virus, Zika virus, HIV virus, Epstein-Barre virus, and cytomegalovirus can also cause the Gullian Barre syndrome. The antibodies are formed against these viruses or these infectious particles, and they do not understand its self pathway, and they attack on the myelin sheaths of the neuron. First, the CMV, Epstein-Barre virus infection, HIV virus infection, hepatitis E virus, and Zika virus infection occur, and due to the reinfection of these diseases, due to reinfection cause the Gullian Barre syndrome. Mycoplasma pneumonia can also, the new reinfection of mycoplasma pneumonia. The reinfection of the mycoplasma pneumonia can also predispose to Gullian Barre syndrome. The Gullian Barre syndrome also occur much more in Hodgkin lymphoma. Gullian Barre syndrome (GBS) can occur much more in the Hodgkin lymphoma. The Gullian Barre syndrome much more occurs in the Hodgkin lymphoma, SLE, systemic lupus erythematosus. HIV positive patient can predispose to Gullian Barre syndrome. Seropositive individual and SLE can predispose to GBS, Gullian Barre syndrome. The Gullian Barre syndrome occurs due to autoimmune pathology, due to autoimmune processes. The autoimmune antibodies are formed in the body of the human, and they attack on the myelin sheaths of the neurons. The autoimmune basis, there is autoimmune polyradiculopathy occur. Autoimmune polyradiculopathy occur. Autoimmune polyradiculopathy occur, in which the cellular humoral immune mechanism both are involved. Cellular immune mechanism and humoral immune mechanism both are involved. The T cell activation occur. When the T cell activation occur, there is elevated level of cytokines and cytokine receptors. There is elevated level of cytokine when the autoimmune process starts, and cytokine receptor are also increased. Cytokine receptor are also increasing. The interleukin 2 increasing. The interleukin 2 increasing. The interleukin 2 are increasing. The interleukin 2 are increasing, and in the CSF analysis of the GBS patient, there is tumor necrosis factor gamma are detected. Tumor necrosis factor gamma are detected in the CSF analysis of the Gullian Barre syndrome. The evidence states that all cases of the GBS there is immune specific antigens are present. All cases of the GBS, the evidence states that all cases of the GBS, there is immune response occurs. All cases of GBS, in all cases of GBS, there is immune response. There is immune specific response. There is immune specific response. Immune specific response are present in all cases of the GBS. In all cases of the GBS, there is immune specific response are present. And antigens are formed against the infectious agents. Antigens are formed in this pathogen are formed against the infectious agents. Antigens are formed against the infectious agents. These antigens mediate to the host tissue. These antigens mediate and goes to the host tissue. They go to the host tissue. And when they mediate to the host tissue, a resemblance of apoptotic mechanism occur. They resemble to the host tissue. Resemblance occur to the host tissue by a mechanism called apoptotic mechanism. By the apoptotic mechanism, they resemble the host tissue. This antigen form against the infectious agents, against the Campylobacter jejuni, against the upper respiratory tract infectious agents, and antigen are formed against the upper GI tract infection, and these antigens are also formed against the infectious particle. When there is infection, the antigens are formed, and the antigens are formed against this infection, and these antigens resembling the host cells, and by the mechanism called apoptotic mechanism, and their resemblance to the host cells. Finally, they target the neuron. Due to this resemblance of the host cells, this antigens target the neuron. In cases of GBS, they target the neuron, specific regions of the neuron, that is called gangliosides. They target the specific region of neuron due to the resemblance to the host tissue by the apoptotic mechanism. They target the gangliosides. The gangliosides contain gangliosphingolipids. The ganglioside contains the sphingolipids. These sphingolipids called glycosphingolipids. They contain one or more salic acid. This ganglioside contains sphingolipids that contains one or more salic acid. This contains one or more salic acids. When they target the salic acids, the various gangliosides precipitates. They attack the gangliosides and gangliosides are broken down. These gangliosides are broken down when these antigens are attacked on the gangliosides, and these gangliosides are broken down. The gangliosides contain glycosphingolipids and one salic acids, and this gangliosides particles are precipitated. This particle of the gangliosides are precipitated. When they precipitate, they are typically exposed on the plasma membrane of a cell, such as node of Ranvier. These gangliosides present at the plasma membrane of the neuron. For example, this is neuron, and this is node of Ranvier. This is present at the node of Ranvier. When these gangliosides and salicylic acid containing glycosphingolipids that contain salicylic acids are broken down at the node of Ranvier, the node of Ranvier become the image, and the impulses will go there. There is no impulses occurs. The transmission of impulses does not occur in case of GBS at the node of Ranvier. The antigens are formed against the gangliosides. and antibodies are found. They are found 20 to 50% of the cases. Found 20 to 50% of the cases of the GBS. The 20 to 50% of the cases of the GBS, the antibodies are found. Particularly the antibody against the acute motor axonal neuropathy, against this pathway, and there is AMSAN. In the AMSAN, the antibodies are found against the AMAN and AMSAN. AMAN and AMSAN, the antibodies are found. And the Clostridium jejuni, the Clostridium jejuni reinfection can also cause the antigen to form, and these antigens are formed, and they resemble the host tissues by the apoptotic mechanism. They also attack the gangliosides. Some autoantibodies react with gangliosides GM1. Some antibodies when are formed, some antibodies attack the GM1 site, in the human nerves. The salic acids from the Clostridium jejuni. This the Clostridium jejuni contains salic acid. These salic acids can also activate the dendritic cells. These salic acids, the Clostridium jejuni contains salic acid. Clostridium jejuni contains salic acid. This salic acids activate the dendritic cell. This salic acid of the Clostridium jejuni can also activate the dendritic cell. And this dendritic cell, this activates the dendritic cell. Due to activation of the dendritic cell, this dendritic cell activates the toll-like receptor 4. This activates toll-like receptor 4. When the toll-like receptor 4s are activated, they promote B cell differentiation. They promote B cell differentiation. When the B cell differentiation occurs, this amplifies the humoral immunity. This amplifies the humoral immunity. The humoral immunity are amplified because of the salic acids of the Clostridium jejuni that contains the salic acid. They activate the dendritic cells, and dendritic cell activate toll-like receptor 4. They promote the B cell differentiation. Due to B cell differentiation, they amplify the humoral immunity. Some evidence state that the IV administration of bovine brain gangliosides, some IV bovine brain gangliosides, some evidence states that the IV administration of the bovine brain gangliosides, when we administer this after 5 to 15 days after the administration of these gangliosides, the anti-GM1 antibodies are found. Anti-GM1 antibodies are found. Some evidence states that these antibodies are found at the node of Ranvier and motor end plate. These antibodies are found at the node of Ranvier and motor end plate. Motor end plate and node of Ranvier. Anti-GQ1B antibodies are found in 90% of the cases. The anti-GQ1B antibodies are found in greater than 90% of cases of the GBS. These antibodies are found, and these antibodies are also found in the Miller-Fisher syndrome. The anti-GQ1B are raised against the Clostridium jejuni. These are raised against the Clostridium jejuni. This anti-GQ1B are formed and raised against the Clostridium jejuni. This GQ1B anti-GQ1B antibodies are formed and raised against the Clostridium jejuni. And they are isolated from the patient with Miller-Fisher syndrome. Isolated from the patient with the Miller-Fisher syndrome. What is acute inflammatory demyelinating polyradiculopathy? Acute inflammatory, what is the pathology in the acute inflammatory demyelinating polyradiculopathy? Radiculopathy. In the acute inflammatory demyelinating polyradiculopathy, there is complement system deposition. Complement system deposition. Due to the depositions of the complement systems, the outer surface of the Schwann cells activate the macrophage. Outer surface of Schwann cell activate the macrophages. This attract and activate the macrophages of the outer Schwann cells, and complement systems are activated in the acute inflammatory demyelinating polyradiculopathy. They damage the myelin sheath and axon. As a result of this, they damage the myelin sheath and damage of axon. Damage of axon occur. What is the acute motor axonal neuropathy? What is the acute motor axonal neuropathy? Acute motor axonal neuropathy. Acute motor axonal neuropathy. In this acute motor axonal neuropathy, different complement systems are deposited. In this, the different complement are deposited. The different complement system are deposited. The deposition of the different complement system occur in the acute motor axonal neuropathy. And IgG are deposited at the node of Ranvier. IgG are deposited at the node of Ranvier. This IgG are deposited at the node of Ranvier. And the antibodies are formed against the GD1. Antibodies are formed against the anti-D1. Against the D1. The antibodies are formed against the D1. Anti-GD1. Anti-GD1. Against the GD1 antibodies are formed. When these antibodies are formed, this damage the nerve sheath. And node of Ranvier becomes damaged. The node of Ranvier becomes damaged, and there is impair in the transmission of impulses. There is impair in the transmission of impulses because the node of Ranvier are damaged and deposited. Autoantibodies are found at the node of Ranvier.